Good afternoon, everyone. So welcome back. So welcome to the section of computational advances. So my name is Xuegong Wang from the Hong Kong University of Science and Technology. So it is my honor to chair the session. So firstly, please allow me to introduce the first speaker, Professor Xuegong Zhang from Tsinghua University. Professor Xuegong Zhang is the pioneer of the field of bioinformatics in mainland China, and he has been working on developing algorithms and computation methods in the field. And recently, he has been working on the foundation model of single cell. And also, he has been recently elected as the vice president of the International Society of Computational Biology. So without further ado, please join me to welcome Professor Zhang to give us a talk.
Thanks, Xuegong, for the nice introduction, and it's a great pleasure to share our recent work and also our perspective for the future of Cell Atlas. So I will start with the two questions that we are interested in about building Cell Atlases. One is how should the Cell Atlas be alike from an informatics viewpoint, how to organize the data. So, so there has been nice metaphors from HCA about the Cell Atlas is like a periodic table of cells, and also like a Google map of our human body of cells. Those are beautiful metaphors, but they do not answer about how to organize the data. As we have seen from the website and also the morning's talk, the current status is still it's a huge archive of data files indexed by study information. This is, of course, valuable, very valuable for the whole community to use as a reference, but I think it's not the final goal of building the atlas. So we proposed that to build the cell atlas should be assembled as a virtual human body that enables doing experiments in the data. So for that assembly, that need to be cell-centric. That means the atlas should be assembled at the cell level rather than the accumulation of studies. And of course, it needs unified information framework and challenging database technologies. So under that perspective, we have built a kind of a prototype for this idea that we call it human ensemble cell atlas based on public data 2 years or 3 years ago. Of course, the size is small. It's only 1 million cells from 38 organs, but it kind of illustrates the idea of a virtual body composed with digital cells. And we invented we call it the in-data virtual drug experiment, and in-data cell sorting, that in the sense that since all the data are organized in one huge file, no matter which organ they are from and which study they are from. So we can use a few lines of computer code to do cell sorting on this virtual body, and using that we can study the possible off-target side effects of some drugs. That's the first version, and recently we have completed the second version of that, which with 10 million cells, also from all public resources, and we have an upgrade UHAF similar to the CAP presented by Chloe this morning. And besides all that work, and we have particularly defined the cell atlas to be AI friendly, that is ready for building the large cellular models. That is about our second question about building cell atlas. That is when we have really accumulated a huge amount of data, and how can we make the atlas alive? That is So in a sense that we have, after we assemble all the cells at cell level to a virtual human body, it is a body, it's not a life, because it is kind of dead. We don't know what will happen if we perturb some of the genes. And so we need to make it alive and by understanding how the genes work in different type of cells. So that is the motivation is can we turn the virtual body to virtual life? And the key to that is we need to understand the cell language, the story of the cells that has been told by the transcriptome, of course, in the future, other omics. So, to tackle that challenge, I think the advancement in AI, especially in natural language understanding using large language models, give us some possible direction for that. We know that the current large language models have been pre-trained on huge amount of text materials, and somehow it masters the language, the human language, and can tell many things. And so based on that idea, we think that we ask that can we use AI or can we design AI models to learn the cell stories, to understand the language of cells, so that we can make the digital cells alive. So that's our, and we had been trying to exploring that on that route from several years ago. This is one of our early efforts that we developed a AI model. We called it Unicorn, to learn the multifaceted relations among the genes. So the first author, Huang, is in the audience, although he has already graduated from our lab. And after that, we built this model. We call it IC Foundation, to learn the genes and their relations in all contexts. We collected 50 million cells from all available sources at that time, 2 years, 3 years ago. And we designed this large cellular model to learn the gene, to try to predict the gene expression from its context. And if we succeed to do that, then we kind of figure out the relations among the genes in different cell contexts. So this is a structure, is a kind of is a modified transformer structure. So for the interest of time, I will not go into the details of this, but there are some inventions in our model. One is the way to deal with the gene expression and to treat the gene expression as kind of a language token. So although we can do the metaphor that genes are like words in the natural language, but they are not actually. So they have different expressions. So we invented this way of to also discriminate the gene expression values in a high resolution manner, so that we can embed the exact gene expression value into the token vector. And based on that, we developed a symmetric transformer structure so that we can deal with all the 20,000 genes at the same time. So that will make the network to be really huge, to be intractable for the current hardware. So we designed this asymmetric transformer to take advantage that many of the genes are actually missed in the current single-cell technology. So we can save the computation there. And also we designed a new pre-training strategy. We call it rate depth aware modeling. That is, when we predict the missed genes, we consider the total sequencing depth of the cell as one of the conditions. And based on that, we, after pre-training in a all the self-supervised manner on the 50 million cells, we get the embeddings of genes, gene expressions, and also the embedding of cells, and this makes the assay foundation can work well on downstream tasks like clustering of cells, annotation, gene module inference, and especially we can infer to predict the response of different cells to different drugs, and also can predict the react of gene expression of the cell after some gene, one or two of some genes are perturbed. So they said in this sense, why people call it a foundation model, because that when we trained the model, we didn't take consideration of any of these particular downstream tasks. But after pre-training, the model kind of learned the semantics of the gene expression and in different contexts, and they can be applied on all the downstream tasks in a way that performs better than the specifically designed models for those particular tasks. So this actually is just one example of the recent advances in this field. And we call it the large cellular models, as Aviv has mentioned in the morning's talk. So there have been quite a few such models starting from the assay board, and later last year, GeneFormer and SAGBT and our assay foundation, and the later Gene Compass and our latest assay Mulan. So the difference among these methods, so the basic idea are more or less the same. We try to use transformer-based structures and use a huge amount of data to pre-train the model, and so that the model can learn to perform on downstream tasks. The difference between those methods are the ways to tokenize and embed the gene expression and the details of model structure, design, and the pre-training task, and also the data sources and downstream ways of application. I think it's still too early to try to answer, to summarize what is the best practice, but all they are valuable efforts. And I had the honor to invite all the authors of these models to write an opinion paper on our journal, Quantitative Biology, so you can find their thinking behind their work. And the long story short is all this work has illustrated that such kind of a pre-trained task-agnostic large cellular models can perform well on almost all downstream tasks related with single-cell biology. Well, virtually almost all, except one task is important, that except the generation of totally new single-cell transcriptomic data. And we have all heard, I guess, we have also heard that Richard Freeman said that what I cannot create, I do not understand. So then we think whether we can create some new unobserved single-cell data. So that is our latest model, assay Mulan, specifically designed for generation. So we combined the gene expression and also the metadata together to define the C sentence or cell sentence, and using the HCA version 2 with data augmentation, and that makes the data to be equivalent to 100 million cells. And with that, we can use the real GPT style autoregressive generation way of pre-training. And after that pre-training, we can do zero-shot generation. Not only, so the traditional tasks like cell attributes prediction, like cell type annotation and so on, so forth, it's a natural part of this generation. And also we can use conditions to generate cells that has not been observed in the training data. And also use that, we can do the zero-shot in silico perturbation so that we can explore what are the possible regulators that can drive the cell type transfer from one state to another state. And eventually we are we're trying to get continuous gradient of some conditions so that we can generate the cellular movies along some developmental or disease genesis process so that we really make the virtual body alive. So this brings out the future of our vision about the future. So currently, as also we also mentioned in the morning, so we actually have proposed this several years ago that our final goal should be to build first virtual cells based on all the single cell and spatial omics technology. And then eventually we will build virtual organs and virtual say embryos and eventually virtual bodies. So in this way we are kind of building the digital version using mathematical and AI models, the digital version of the carbon-based life. So we call it the digital life. It's not digital body. So it's a digital life. And in Chinese, we call it 数机 life because the 数 in Chinese, not only is digital, it's data, but also it is mathematics. So we can use both the simple and also the complicated AI mathematical models to kind of to reproduce life in the digital space. So I had written this perspective of building the digital life for future biology and medicine. I think with this I will stop here and will be glad to answer questions. So all these people, some of them are in the audience today. So it's a big effort from our lab. So, yeah.
All right, thank you very much for the very nice talk. So any questions from the audience?
Thank you, Professor Zhang. Very, very exciting talk. So with the emergence of foundation models, I'm actually very curious how much we are getting closer to understand the gene regulation. And can you comment on that?
Yeah, that's a good question. So I think so we have to realize. Actually, we also, as we discussed in the lunch meeting with 10x people. So we have to realize the current data are not perfect in terms of all the possible regulation. So the number of genes we can measure. Both for single cells and for spatial transcriptomics, they are limited. And also there are many, say, non-coding genes, microRNAs, that they are not, they're virtually ignored at the current stage. But given, keeping that in mind, for those genes that have been observed, I think the foundation models are performing pretty good for gaining the gene relations, not necessarily causal yet, is still association, but especially for the highly variant genes. So the current strategy provides really powerful tool to capture the relations that can sometimes be ignored in some minor cell types. So that's my general question.
Thank you.
Hi, Professor Zhang. It's a really nice talk. And actually, I don't have a question, but I just want to hear your insights or comments. So for this kind of foundation model, you probably will give us like kind of low resolution cell type annotation, for example, for CDA versus CD4, right? But sometimes for biologists, we might need like higher resolution cell type annotation. So I want to hear your comments of how to combine this kind of foundation model with,
Yeah, that's a good question. So the current published foundation models, the cell type annotation is kind of at low resolution. I agree with you. But what I say Mulan does is that we are producing the hierarchical annotation, And we have the U-half, the unified hierarchical annotation framework. So we are trying to design a hierarchy of cell type and cell states. And I say Mulan at least has improved the resolution. And with the future accumulation of more and more data and more coordinated annotation at multiple resolutions of the cell type, I think this will, so the large models can work on detailed, more fine resolution definitions of the annotation.
Xuegong, you brought a very interesting point about the difference between models that fit some data, say in an autoencoder sense, versus models that are causal. And I'm intrigued by the fact that a lot of the excitement around foundation models for single cell is around virtual cells, as you mentioned, and being able to predict causal effects of, say, perturbing one gene. I want to get your thoughts on, can the existing methods really do that if they're not trained on knockout data?
Right.
If you're just observing lots of transcriptomes, can you really predict the
that's
perturbations in any non-trivial way?
Yeah, that's a very good question. And actually that question is, I know that some people don't believe, or at least they challenge the possibility. That's because the current large cellular models, although including our model, so we have succeeded in some experiments showing the performance, but if you do some other performance experiment, we are also collaborating with Huang on that. So the performance is not that satisfactory for the unseen perturbations yet. But that can be, so I think it's the current phase of this large foundation models is kind of still at its infancy, is at very early stage. So because our data are not that complete yet, and the resolution of data and noise data level is high. So with I say Mulan, we get some improvement, and also we are collaborating with the Huang on further improvement. So the short answer is that it is not close yet, but it is really promising that we can see when we have more and more data, although it's not directly the perturbation training data, but we have seen all the different possible combinations of gene expression in nature, then we will be able to, the model will be able to predict what will happen if something not being observed in nature happen. I think that we are more optimistic for that. But only we need to be more patient and waiting for the quality and quantity of the data, and also the power of the model to increase, to grow up.
Thank you.
All right, thank you very much for the nice talk. Please join me to thank Professor Zhang again for the wonderful talk. Now I would like to invite our second speaker, Boxiang from National University of Singapore. Boxiang is a PI and director of the Genomics Data Science Lab in the NUS. His research group has been aimed to understand human complex disease through genomics, single-cell, and spatial transcriptomic analysis. So please join me to welcome Professor to give us a talk.
Okay, thank you very much. It's my pleasure to be here. My name is Boxiang, and I'm from National University of Singapore. And today I'm going to talk about a new tool from our lab called DIRAC. And this work is done by an extremely talented postdoc on the top right hand side from Zhang. And he's also in the audience today presenting a poster. So if you have any specific questions about this tool, I encourage you to contact him after this talk. So because we're in Human Cell Atlas meeting, I want to point out that two of the key goals of Human Cell Atlas is first to build complex atlases for multiple tissue and multiple cell types. And the second key goal of the Human Cell Atlas is to go multi-omics. So most of us focuses on transcriptomics, but as many of you know, we have genomics, chromatin accessibility, and metabolomics. And here is just a screenshot of some of the papers that were published recently for spatial multi-omics, including spatial site-seq, spatial cut and tag-seq, and then stereo site-seq. So facing with these two goals, we have two fundamental challenges. The first one is how to align cell type annotations across data set to build large-scale atlases. And the second challenge is how to align genomic features across multiple omics modalities. So here, before I dive into the result, I want to talk to you about the framework that we're thinking about these problems. So there are two prevailing paradigms for integration. The first one is what we call horizontal integration, basically transferring labels across data sets. So here, each row, each column represent a batch. So here, batch 1, 2, and 3. And each row represent a different modality, right? RNA, attack, and protein. So if we integrate across these batches, our key goal is to remove batch effects and align cell type specific annotations across data sets. And these integration uses genomic features as anchors. And one example is that we can do cell type transfer from reference data set to a query data set, right? And the reference data set is, of course, annotated, and the query data set is unannotated. So along this line of work, there's multiple methods. For example, scMap, Harmony, Scanorama, Surat, and ScanVI. And on a spatial paradigm, there's one method called Stellar that was published 2 years ago on Nature Methods. There's a different paradigm of integration, which what we call vertical integration, right? So this is integrating in the same batch, but across different modalities. So the key goal here is to remove batch effects and align multiple omics modalities. And so here we don't use genomic features as anchors. Instead, we use cells as anchors, or spots if you were talking about spatial transcriptomics feature. So an example here is the joint analysis of spatial attack and RNA-seq data. And of course, there's multiple methods that performs this task on a single cell level. Most well-known, Liger, INMF, GLUE, and Multi-VI. And on a spatial level, there is a new tool that was published earlier this year called Spatial Glue that specifically is designed for this task. So here I just summarized the method that I talked about before. So horizontal, there's these methods. And vertical, there are these methods. And then there's also another task which is called bidirectional, right? A method that does both and does both well. So for single cell, there is one method which many of you have used, is Surat version 5. But there is no such tool that does bidirectional integration for spatial data. Okay? And that's what DIRAC is going to address. So before I dive into the result, I want to shift our mindset a little bit. So both integration, vertical integration and horizontal integration are special instantiations of a much broader class of problems called domain adaptation problems, right? I like to motivate this using an analogy from computer vision. So suppose that we're trying to tell between cats and dogs. And then horizontal integration is basically you're doing the same task, but using a different white balance, right? So on the right side, maybe your background is not white, your background is light green. This is what we call horizontal integration in a computer vision point of view. And vertical integration is basically you're measuring the same object, but using different sketches, right? So perhaps a cartoon depiction of cats and dogs tells you the same semantic information, but they depict the same. They depict a different contour of the same object. And as I mentioned before, both of these are special instantiations of a much broader problem called domain adaptation. And I like to quote a paper from 2016. For effective domain adaptation to be achieved, predictions must be made based on features that cannot distinguish between the source and the query. So basically, if we want to build an effective model that performs all these tasks, we have to remove the features that are domain-specific and only keep the features that are cell-specific, right? So we keep the features that describes the cell types and cell states and remove the domain-specific features. So that's exactly what we did. This tool called domain invariant representation by adversarial calibration. And I'll explain to you why we call it adversarial. So the first step is very simple, right? We construct a graphic neural network, and then we use these extracted features to do prediction, right? So the predicted labels are the normal labels that you would use in any task. So for horizontal integration, the prediction can be cell type labels, and then for vertical integration, the prediction labels can be reconstruction loss, right? But then we have another module, which is the domain adversarial discriminator. So what we do is that we predict the domain labels that can distinguish between these omic modalities and also different batches. And then we feed this label back into the feature extractor, and we guide the feature extractor to ignore these features, right? So that's the basic idea of this platform. And I'm going to show you some of the benchmarks that we have done using this DIRAC framework. So the first benchmark we did was on mouse embryonic day 10. And here we used a UDA-seq data. It's a microfluidic-based multi-omics data, RNA plus protein. And the resolution is about 10 microns. So here our task is to vertically integrate RNA and protein. And then we have to do spatial domain segmentation in an unsupervised fashion. So here, again, is the ground truth. This is the manually labeled ground truth. And this is SURAT. So obviously, this is not a very fair comparison because SURAT does not take into spatial information at all. So you can see the performance of SURAT is not that good. You see a few spotty regions here and there. And also, the other thing is that SURAT does not use embeddings. So let's say we first add embeddings. Let's say we use multi-VI, right? And as you can see, multi-VI's performance is slightly better, but the key problem with multi-VI is that the embeddings are separated. They're not well mixed, so they don't address the domain separation problem. What about if we use both alignment and embedding? So let's say we use MaxFuse. So MaxFuse perform better than multi-VI because the domains are now well mixed, right? But the problem with both multi-VI and MaxFuse is that they don't use spatial information. Okay, so let's say we use spatial information plus embeddings. So here you can see spatial glue. It performs much better than the previous three methods, but still, you see a line at the top, which is not, is not supposed to be there. And the problem with spatial glue is that even though it uses spatial information and embedding, it does not correct for the batch effects, right? So let's say you do all three. You do alignment, you do spatial information, and you do embedding. Now you have DIRAC. Okay, so here you see that DIRAC corresponds extremely well to the ground truth, and then the omics are extremely well mixed. A slight digression, if we don't have spatial information, DIRAC also performs on single cell data. So let me just show you a few examples. So we test the method on adult mouse cerebral cortex. So it's a single cell measurement of RNA plus chromatin. And then we have manually labeled cell types based on marker genes. So here our task is to integrate RNA plus chromatin accessibility to annotate the cell types in an unsupervised fashion. So here, we're comparing a few methods here. So this is UniCom, apparently does not perform well at all. And then this is SURAT. It performs well in terms of omics mixing, but not, but it's okay for biological preservation. And this is multi-VI. It performs very well for omics mixing, but it does not perform well in biological preservation. And both GLUE and DIRAC performs very well on biological preservation and omics mixing. So we compared against 15 different tools. It's really a big effort by Xuegong, and DIRAC performs the best for both biological mixing, biological preservation and omics mixing. So I'm going to show you the horizontal integration benchmarks. So let me just set up the context first. So here we're using the human prefrontal cortex as the benchmark. So this is done by 10x Visium RNA dataset, and the resolution is about 100 microns. So here what we're going to do is to use a reference slide, manually annotated, to do horizontal label transfer and to annotate the query slide. So I'm going to show you, again, from the worst performing methods to the best performing methods. So here we performed against a very strong baseline, which is SVM, and also a method that's specifically designed for horizontal label transfer called SCMap. So both of these methods missed important layers, missed layer 2 and layer 4. These are the two difficult layers, apparently, for human prefrontal cortex. And now we also have Stellar, which I mentioned before. This is specifically designed for spatial omic horizontal label transfer, and AdaBoost, which is a strong baseline, and also ScanVI. So all these three methods can detect all 6 layers, but the boundaries are very fuzzy. And now you see DIRAC, which not only detect all 6 layers, but the boundaries are very sharp, right? And it corresponds to the results extremely well. DIRAC performs so well, we decided to handicap it by removing labels from the reference data set. So one advantage of DIRAC is that not only can it use labeled data as the reference, it can use a mixture of labeled and unlabeled data, which is a huge advantage if you have a ton of data like Human Cell Atlas. So here, this is DIRAC unhandicapped, healthy DIRAC, but these two are DIRAC handicapped, right? So the second column to the right is DIRAC using 70% of the labeled data. And then the third column to the right is DIRAC using 20% of the labeled data. Even with 20% of labeled data, DIRAC outperforms other methods using 100% of labeled data, okay? And then also we can do cell type annotation using Kodak's protein imaging data. So this is the ground truth. And DIRAC result is almost indistinguishable from the ground truth. And we compared against 6 other methods, and DIRAC performs the best. So last but not least, I'm going to bring your attention to some biological insights that we can extract using DIRAC. So here we're using mouse thymus as an example. So we did DAPI stain and stereosite-seq. And we can use DIRAC's vertical integration module to do spatial omics segmentation. And indeed, we segment all 4 of the major spatial domains: capsule, cortex, CMJ, and then the medulla. And if we do bin50, which is difficult for some methods, and not a problem for DIRAC at all, we can detect more fine-grained spatial omic domains. For example, capsular regions, cortical level 1, 2, 3, and medulla level 1, 2, 3. So then we move on to the horizontal integration capability of DIRAC. So here we use a single-cell CySeq dataset as our reference. And then we use DIRAC to horizontally annotate the stereosite-seq data. So the first thing we did is that we extracted the developmental trajectory of T cells in the thymus, because T cells matures in the thymus. And we can actually map the spatial distribution along the T cell trajectory. So here you start with the entering thymocyte progenitors, and they become double negative cells, which moves from the CMJ to the capsule. And then these double negative T cells become double positive T cells, which fills the cortex regions. And then they become single positive T cells that moves to CMJ. And eventually these becomes mature CD4 and CD8 T cells that moves into the core of the medulla. So I encourage you to talk to Xuegong after, during the poster session, but just to summarize the results, bidirectional integration is key to establishing spatial multi-omics organatalysis, and there's no existing method to perform both spatial multi-omics integration in a bidirectional fashion. So DIRAC unifies horizontal and vertical integration as a domain adaptation problem. It outperforms 15 existing methods for these integration problems. And also we provide a resource for spatial multi-omics atlas of T-cell development in mouse thymus. And these data are not published just yet, but we're hoping to write up a paper soon. So just sort of, we're recruiting postdocs and students. So what we do in the lab is occasionally very good science, but more importantly, we have fun. And if you're interested, you can contact me using this QR code.
Excellent talk. So questions from the floor? Right, probably I have one question. So you're talking about the multi-omics integration. Do you also consider the imaging feature, for example, the H&E, the pathology, like cell morphology?
Yeah, very good question. Thank you very much. We have published on that, actually. There is a method, you can, actually Xuegong is the first author of that method to integrate spatial information with the H&E stains, yes. Maybe we can have them talk a bit more about that later. Yeah.
All right. Thank you.
Boxiang, very interesting large-scale integration. As you know, we're generating massive data sets.
Yeah.
One place where I think spatial makes life hard for all integration algorithms is that there are too many cells now. You can get to many, many millions of cells very fast. So I'm curious, comparing all these methods, including DIRAC, how can they handle like 10 million and in future tens of millions of cells?
Yeah, that's a great question. So for some of the methods that were performed, that we compared, they do not scale, but we have designed DIRAC specifically to scale to millions of data. We have tested on up to 2 million cells, and it can perform integration within less than 1 hour. The way we do it is that we use graph sampling, right? So if you think about this problem, your memory explodes because you're putting a graph into the neural network. But if you use sampling techniques, then it scales much better.
Actually, a lot of neural net methods just sample the data to handle scalability. Just when you comment, I mean, intellectually it feels like you're not using the full value of having a massive data set if you just sample it. I'm curious to get your thoughts on that.
Yeah, so it depends on the range of interaction that you have, right? So I'd say that it's a common negative to use neural networks because in order to generate these embeddings, you have to consume a lot of memory, right? So it's a trade-off. I mean, we can tune the size of the sampled regions, but in our hands, either sample or no sample, the results are very similar. Yeah.
Okay, great.
Yeah.
Great talk, Boxiang. Question about, so can bidirectional integration alleviate some of the problems we have with segmentation, where are you able to leverage this aspect to better call and deconvolute some of these cells which may have been in contact with other cells nearby?
Yeah. Segmentation is a super hard problem. I'd say that's like the fundamental challenge that has not been solved. I mean, there's at least 3 methods doing segmentation on subcellular resolution data, but I don't think the problem is far from solved. So one thing you could do is that you can iteratively use DIRAC and these cell segmentation methods to improve the boundaries, right? You expect that molecules from the same cell to behave like they are from the same cell. So you can adjust your boundaries that way. But here, I think the challenge is the computation. But I think you're absolutely right, is that we can iteratively refine the cell boundaries using using both tools. Yes.
Last question.
Oh, hi, Professor Liu. It's a really nice talk. Actually, I checked your paper on the GitHub, and seems like you guys use the graph neural network for both RNA tag and different modality.
Yeah.
So, do you consider using different framework for different modality of the data because they have different features?
Yeah. So we the nice thing about DIRAC is that it's one framework to rule them all. So it's basically domain all these problems are special instantiations of domain adaptation. So if we build a framework that's tailored towards domain adaptation, you basically fit all these problems into the same framework. So we do not have to, I mean, we could, but we do not have to use different frameworks for different modalities or different batches or different technology platforms. Just use the same framework.
All right, so let's join me together to thank Professor Liu again for their nice talk. So let's move on. So let me introduce the third speaker of this session, so also the last speaker, Dr. Drew Neavin. So it's a postdoc associate in Garvin Institute of Medical Research. And I think she will give us a talk on the genetics and single-cell technologies. Please.
Perfect, thanks. Good afternoon, everyone. I first want to start by thanking the organizers for giving me the opportunity to talk about this project, which has been going on over the last couple of years, and it's finally coming to a point where we're able to pull out some really interesting information from the data and understand the distribution of diversity across single-cell atlases. And I want to start by acknowledging, if the pointer goes. Can you advance? Perfect, thank you. I want to start by acknowledging the traditional owners of the land in which this work was carried out, the Gadigal peoples of the Eora Nation, and I want to pay my respects to their elders past, present, and emerging, and welcome any First Nations people joining us here today. All right, so I want to first start by talking about some different terminology that's sometimes used interchangeably, but I want to differentiate it here for this talk. The three terms are ancestry, ethnicity, and race, and actually ethnicity and race are socially ascribed entities and concepts, whereas genetic ancestry is really based in biology and allows us to understand the genetic origins of an individual's genome. And we can do this because of the historical migration patterns of humans across tens of thousands of years that have given rise to different allele frequencies in different populations, which really allows us to understand the genetic origins of an individual's genome across this landscape. And I think you've seen this slide already once today, but I just want to point out that although we know that diversity is a really important contributor to inter-individual variation, historically, especially in genetics research, we really aren't capturing that genetic diversity with our studies. And so this is a publication from a couple of years ago of genome-wide association studies, where about 90% of the studies to date are in European ancestry individuals, but you can see on the right there that this is not a really representation of the world. The global representation of European ancestry individuals is about 15 to 20%. And so this is drastically different from the studies that we're carrying out. And so there's a lot of work in the community trying to rectify this and get this to be more representative. And in single-cell data as well, we know that there are differences that we can pick up from single-cell data across different ancestries. And here I'm showing you an example from Luis Barrero's lab, where they looked at genetic ancestries of European and African ancestry individuals and show consistent differences in gene expression in PBMCs that have been treated with a viral stimulus. And they were able to then show that these changes are also really related to variation that we're seeing in COVID-19 severity. And so not only is this a gene expression profile thing that we can pick up, we can also see that it really translates into the clinic and can impact how we actually are treating patients in the clinic as well. And so we know that diversity, and I think this dovetails really nicely after this morning from Shyam's talk and some other of the introductions where we're thinking more about diversity across the human cell atlas. But historically what we've been doing is pooling together information, but we haven't been really accounting for what the diversity of ancestry is in the single cell atlases. And so we really set out to quantify this. And first we had to set up a method to help do this. And so we chose two different ancestry methods that we could use to understand genetic ancestry across samples. So the first is using genetic admixture, and so this allows us to take an individual and look at the proportion of their genome that can be attributed to different ancestral regions across the world. This is typically done at a continental level because it's very challenging to get to more resolution than that. And so here you can see this individual on the right has about half of their genome that can be attributed to African ancestries and a quarter each to European and Indigenous American ancestries. We also decided to include a principal components method, which is a little bit more quantitative than this admixture method, and allows us to look at the genetic ancestral similarity between two different donors. So a little bit different from what you're probably used to looking at with a principal component plot. Each point on this plot is a different individual as opposed to a different cell. And so typically we would do these types of analyses using whole genome sequencing data or SNP genotype data, which are kind of our gold standards for the field. But as you might imagine, whole genome sequencing data has a lot of different characteristics to single cell data, and most of our single cell data in our atlases actually don't have SNP genotype data or whole genome sequencing data available, which means we need to leverage the single cell data itself to be able to understand the genetic ancestry of the samples that have been included in the atlas so far. And so here you can see that we really don't get good distribution across the genome compared to whole genome sequencing data with single cell data, and it's much more sparse than whole genome sequencing data. So these are two things that we had to really keep in mind as we were trying to identify a method that we could use to estimate genetic ancestry across single cell data. And so if we take a look at genetic variants here across the genome, what we can see is that specifically for variants that are overlapping regions that aren't assayed by a single cell assay, we won't be able to detect those in single cell data, whereas we would with whole genome sequencing data. If we then look at a region that is covered by a single cell technology that we're looking at, we might not have the depth to actually be able to detect the type of variant that we would usually be able to detect with the whole genome sequencing data. And so these are two things that we really had to keep in mind with developing this method. And so the culmination of this work has been the development of single SC Ancestry, which is a scalable, reproducible, and accurate method for estimating both admixture and principal component analyses. And we've built it in a way that it uses a reference so that the estimation of the ancestry can be done in multiple different labs and directly compared to one another, making it very easy to look at samples from multiple different locations. And so to do this, the first step is to call genetic variants from the transcriptional reads or from the attack reads or whatever single cell technology you have. And then we phase and impute those variants so that we can correct for variants that don't have very good sequencing depth, followed by filtering for high quality variants and removing variants that are in tight linkage disequilibrium. And then we're able to use these genetic variants that are called after all of these steps to estimate admixture and principal components for ancestry. And so we tested this using that same data set that I introduced in the earlier slides from Luis Barrero's group. And the reason we use this data set is it has paired whole genome sequencing and single cell data from a diverse data set of both European and African ancestries. And so when we apply SC Ancestry comparing the whole genome sequencing admixture estimations on the y-axis to the admixture estimations for the same samples using the single cell data with SC Ancestry on the x-axis, we see a very strong correlation indicating that our estimations of admixture from our single cell data are very reproducible with what we would typically get with our gold standard whole genome sequencing data. And this is very consistent with what we see for principal components as well. This isn't a perfect map of whole genome sequencing to single cell data, but we do see that with SC Ancestry, we're able to capture the global distribution of individuals into these different regions of the principal components areas. All right, so having developed this method that we were quite happy with could help us understand genetic ancestry of single cell atlases, we've applied it to a number of different atlases that I'm showing you here on this slide that comes to about 15,000 different samples. And while we're still processing some of this data, we've to date been able to process just about 7,000 samples that covers 153 data sets, 17 technologies, and more than 18 million cells. And you can see that most of these are from HCA itself, with a large overlap with CZI, and then about 500 from the HTAN network as well. And this is kind of the foreboding conclusion. We have about 77% European ancestry samples with at least 90% of the genome of European ancestry in the samples that we've looked at so far. And so this is a pretty large proportion of bias towards European ancestry samples, with smaller percentages of other ancestral regions as well, and about 18% that are admixed between different ancestral regions. On the bottom, you can see our PC plots with all of the samples on the PC on the left, and then a density plot on the right so that you can see that most of the samples are in the bottom right-hand corner where we have our European ancestry reference samples. And so since we're at HCA Asia, I wanted to point out some contributions to the East Asian and South Asian ancestries. And specifically, we know that a lot of the data that has provided a higher percentage for the East Asian ancestries comes from the IDA data set. So about 60% of the East Asian or South Asian samples comes from the Asian Immune Diversity Atlas. And so they're making a really big contribution here. But that's only in blood. So when we take a look at blood compared to the global population, we see that the East Asian proportion is very similar to the global proportion, but we're still missing a lot of other ancestries here. And when we move to look at other tissues as well, we see that this distribution is still really quite biased towards European samples. And so this is the point at which I say, I really hope that as a community, we can start moving towards building diversity into our atlases and thinking about just building diversity into any experiments that we're designing so that we can move away from having this large bias of European ancestry samples. So I hope I've been able to convince you that SC Ancestry is an effective method for estimating ancestry across single-cell data, and we'll be making this as a tool that's available for the community and have an interactive method for looking at the results from ancestry estimation across all of the different samples that we've processed. And we see this really strong bias, but I think some of the good news is that there's a number of initiatives undergoing already that are focused on bringing diversity to these different atlases. A lot have been funded by the CZI Diversity Network, but I don't think we should stop there. I think we should all be thinking about how we can really expand diversity in the studies that we're carrying out. And so in the future, what we're going to be doing is using the genetic ancestry and other measures of biological variation to understand how they're impacting gene expression variation. All right, so with that, I'd like to thank everybody who has contributed to this project. It's been really fun working with everybody on this slide who has brought really interesting expertise to the table. And I want to shout out to Elaine Chu from the Chan Zuckerberg Initiative, who's been my partner in crime the last couple years, helping really push this project forward. So thanks for your attention. Happy to take any questions.
Wonderful talk. So questions.
Hi, Partha.
Drew, two quick questions. One is, in your experience, what proportion of the whole genome, what proportion of variants that you cover in the whole genome are actually covered in the single cell? Question number one. Question number two is, even if a variant is covered in the single cell data, not all cells will have that particular variant. So what do you do in that context?
Both really great questions. So we found that the number of variants that are called really depends on both the technology as well as the sequencing depth. So those range from anywhere from about 20,000 variants to over 1 million variants per individual, but it really depends on the technology that you're using. For a typical 10x data set, it's on the range of 100,000 to 200,000 variants per sample. And we look at genetic ancestry across all of the cells in a given sample, and so we effectively pseudo-bulk it, and so we're not looking at individual cells, which allows us to get beyond a little bit of the sparsity, not fully entirely beyond the sparsity, but it does help with that, and so we're not doing individual cell-level ancestry estimation.
Oh yeah, I have a question here. So basically, this is a very interesting story. So I'm wondering how much affected the SC ancestors applying with similar strategy to estimate the effect of like the genetic to kind of linking to diseases like diabetes and all those that well-known questions that people study in the genetic field.
So you're asking how SC ancestry can be applied in diseases to?
Yes.
It sounds kind of like a polygenic risk score question.
Yeah. Yeah.
So yeah, this method really isn't meant for polygenic risk scores. It's really meant for understanding ancestry, but it would be very useful if you're looking at different diseases across cohorts. because we know that genetic variants in different populations can influence then our conclusions if we're not accounting for that effectively. And so this will allow you then to know what the distribution is of ancestry in, say, your cohort or another publicly available cohort that you're using and correct for that effectively.
Yeah, but actually my question is how much it can be effectively, like people using the whole genome sequencing to evaluate the effect of the disease, or how much the genetic variants could, contribute into the disease. So how much is SC ancestor could replace the whole genome sequencing? I mean,
you mean for ancestry estimation? It's
not ancestry estimation. It's just the disease, vulnerabilities and those type of things.
Yeah, this tool really isn't meant for looking at disease-based estimations. There are some other tools out there for single-cell data that are looking at more of that like polygenic risk score-based method for single-cell data and trying to understand disease likelihood, right, in an individual. But yeah, this method just really isn't meant for that. But thank you.
Okay, thank you.
Last question.
Yeah, great work, and thanks for highlighting IDA. I think there are two aspects. One is I think we can do a lot better in data sharing. So I'm just wondering whether there's something that we can discuss in the next 2 days, is what kind of efforts we can make to really ingest and provide the data to HCA community. Because I surely know there are lots of data being generated in this part of the world. And
yeah.
if we can contribute, I think we can really reach that level of equity. Yeah, I guess the second point is, I forgot what I was going to ask.
Yeah,
but yeah, I think it would be great to have more of a comment, to have the whole community contribute the data to the.
Yeah, I think that's a really good point. And one of the things that we're working on is mapping the individuals to location as well, so that we can understand the genetic ancestry across the globe that's being collected. But I think another thing to keep in mind is most of the data that we're using so far is publicly available. And so we may be biasing the distribution slightly because we're only pulling mostly from publicly available resources. And so I think you bring a very important point here that we probably need to be keeping that in mind as well when we're interpreting these results.
I remember what I was going to, it's big and really quick. Because I think now we're discussing about spatial, and a lot of these methods are also moving into probe-based. Just wanted to hear your thoughts on how we can quantify ancestry once we move into that direction.
Yeah, that's
can we do this?
a great question. It's really hard. So I've effectively taken as many technologies as I can for this project, but I can't use 10x flex-based systems because it's a probe-based system. We don't have genetic information there. And I know there are additional methods out there that will have those probe-based systems, which will be challenging. One option that we've been thinking about is how can we train a model that would predict ancestry that's not using genetic information, but using transcriptional information. I haven't worked on that yet, so I can't tell you if that would work or not, but I think it's one option that we can think of to be able to ingest additional types of data that we really can't look at for genetic variants.
Thank you very much.
Thanks.
All right, fantastic. Nice discussion. Please join me to thank the speaker again. So our chair have an announcement, please.
All right, we get started. This session, in the next talk, it's going to be given by Dr. Jonah, who is going to talk to us about immune systems accelerating research through interinstitutional collaborations and all of that. Dr. Kuhl has been very, very helpful to many of us. He's championed our cause and has gotten us money to get started in IDA and many other projects as well. He has a deep love for cell biology, and in particular the origins of cellular heterogeneity and how diverse cells assemble into complex tissues. This originated from his PhD, which he did at the Duke Medical Center, where he was an American Heart Association fellow. He focused on the role of vascularization during cell differentiation and organ morphogenesis. Subsequently, a Ruth Kirstein fellow at the Salk Institute studying nuclear organization during stem cell differentiation. Dr. Kuhl presently works with the Chan Zuckerberg Initiative. He's a senior science program officer on the cell science program of CZI. Earlier, he ran an industry research group working on therapeutic application of 3D bioprinted tissues, and also had worked on intellectual property litigation. So he has a very diverse background, but again, his heart is in cell science research, and that's the reason why we love him, not just for the money, but for the support that he gives. Jonah.
Great, thank you. Thank you for the nice introduction. I'm glad to hear you love me more than for just the grants. I love being here. This is one of my favorite meetings every year, and it's really been a treat to see it grow and evolve and sitting through some of the sessions. So this is a very long title about immune cell research and institutional collaboration. One of the artifacts of a long plane ride and also a little bit of jet lag is I will talk some about immune cells and cross-collaboration, but I also managed to change it up based on some of the earlier talks. Okay, so let me first start very quickly in just kind of summarizing the work at the Chan-Zuckerberg Initiative, and then I'm going to jump straight into science. Just to highlight that there are three major focus areas. So science is really the largest one at this point. Our co-founder, Priscilla Chan, her husband, Mark Zuckerberg, who founded CZI, increasingly refer to us as a science-first organization. But just to highlight that we are also really rooted in education and community. And this is in support of, kind of a number of different efforts, but really the mission statement of our work to support science and technology to cure, prevent, or manage all disease by the end of this century. And the way that we do this is a little bit unique. And I raised this, again, to share, and this will be familiar to some, but I will also try to highlight the kind of unique way that CZI approaches some of the problems in science, in education, and in community, which is a kind of multi-pronged approach. We, of course, fund research around the world. Many folks in this room and elsewhere across the Human Cell Atlas Consortium that people have heard some of their work about today have received grants from us. To support and complement this, we also build technology tools. We've heard some about cell by gene. I'll come back to that and share a couple other tidbits. So this is a really nice example of the work within my group, both to fund single cell research in Human Cell Atlas, but then also build open source tools in ways that that work can be catalyzed. And then lastly, we do work, and this is a growing part of CZI that includes work in our biohubs and institutes, and then also increasingly now a real focus on AI. And I'll end with a couple examples of this and how it ties to the other work. So what I really hope to leave you with is an example and some highlights. Again, very, very brief talk, although my timer's not running, which I take to mean I get even more time. But some highlights across this range of work and related to what I hope are resources that you all can use, as well as ways to plug into these different efforts to best leverage the investments and commitment to HCA and single cell biology that we've made. Okay, so let me reiterate the science mission. So it is to support science and technology to make it possible to cure, prevent, or manage all disease. This came up in the breakout session that I was just in, and we'll simply kind of reinforce that our real focus is on fundamental science and technology. And the view is that by investing in these areas like the Human Cell Atlas, we are not necessarily looking to focus, like many funders, on a single disease or a class of diseases, but trying to generate resources that will help you all do that work, but kind of rise or lift the tide and make it possible and easier and faster to think about all disease. So it's really kind of a force accelerator. And my group is the Cell Science Group. So this is one of the programs at CZI within CZI Cell Science. It's the oldest program, started more or less the same time as the Human Cell Atlas. And the Human Cell Atlas was a really, really important program and project for me personally and the team. Something that our prior head of science, Corey Bargman, and now Steve Quake, who was on the organizing committee for a period of time, referred to as a lighthouse project. And the way that we're thinking about advancing this work more broadly, and also within the Human Cell Atlas, of course, is both accelerating the single cell methods and technologies, and now starting to think some about different applications. And here, AI is a really, really important and exciting avenue. And there was some discussion earlier about different foundation models. I'll come back to this, but this is an area in which we are both now starting to work collaboratively with the community to provide resources in the form of data and compute and benchmarks and others, and also start to train these models and try to work towards this virtual cell vision. So a really important area that we're looking to support and work with you all on. And then finally, to create new technologies to characterize and bioengineer cells. And in particular, the immune system, I think, is a really exciting example of this. And this, of course, relates to current methods like CAR T therapies, but also more broadly to start thinking about how we can take some of the information in atlases and go from reading to writing and starting to write fates and write behaviors or even write histories of cells. And so this is something that I won't dig into quite as much, but is absolutely a big interest. Now, to kind of go back in time, a great example of how we have funded work, and you've heard a lot about this, so I won't dwell, is the IDA project. This was one of the really early bets that we made within originally the Seed Networks program. It then kind of expanded in Ancestry networks, and I think has been a shining star that has taught us a lot and taught the community a lot, but also informed a lot of the other networks and the approaches that we've made. And so, again, won't dwell here. But a great example of how we have supported work across countries and provided kind of catalytic funding to help individuals and groups work across those boundaries and kind of inter-institutional collaboration that I mentioned. Now, one of the key outputs, of course, of IDA and others is our data sets. And here I wanted to spend just a couple quick moments, and Chloe did a great job of highlighting the way that Cell by Gene, which was developed at CZI, is providing support and serving as the matrix store, and a really important aspect of supporting the community in bringing together data in an interoperable and harmonized way that can make it easier for everyone to use. And there's a couple things that I would just add to the earlier work. So one is this is a data resource that is open for contributions. It has to meet that minimal schema, so there has to be associated metadata. The other thing that I would highlight is, and Xuegong framed this really nicely, this is actually a cell-level schema. And there's a couple of things that are really important about that. So there are, of course, many data sets and collections, so you can explore individual collections, often papers. But if you also want to explore all of the cells of a particular type across the entirety of the corpus, the tool will allow you to do that. And it will allow you to do that in a very performant and easy way. The other kind of key use case here, of course, is grabbing all of this data from the census, which is currently a subset of about 70 million cells worth of data. And you can see at the top there, there's an API for access to the census data, as well as now a tab for models. And what you can do with the API is go in, and if you are interested in, again, just a certain subset, whether that be a subset based on ancestry, based on cell type, based on any of the kind of standardized metadata, in a matter of seconds, you can subset that 70 million cell object to just the data you want. There's PyTorch data loaders to help you load it and start to work with different deep learning frameworks and really kind of dynamically use that entire data corpus in addition to kind of the exploration. The last thing I'll just highlight is the models, where all of those examples that Aviv gave earlier were all trained on data that was kind of consumed and used within the census, and that's something that we really want to enable. But of course, the data here is growing extremely quickly. I went on earlier today, and already the slide that I made yesterday is out of date, so I think it's now 102 million cells, and so it cannot keep up. The thing we're doing in a number of cases in models that are widely used is supporting the retraining, right? So that all of that compute does not have to fall onto the labs. And so you can now go on, and if you're excited about GeneFormer or SCGPT, we are supporting retraining, and you can actually access those models via that API in addition to the data. So it's a great way to kind of grab those and fine-tune them or explore them or use them in other ways. Okay, now that was a quick sense of what Cell by Gene is, but I also want to highlight what it is not. And that is that currently it has a very heavy bias, and I always am very appreciative of Cheyenne pointing out some of the biases as well, because I think there's an incredible amount of progress that's been made. There is an incredible amount of work that needs to be done still. So we have a clear bias in terms of the tissues, and that is, of course, going to bias the sorts of diseases and other areas that I know people are thinking about. Within the technologies, it is multimodal, so spatial data is supported. There's still a pretty heavy bias in human and mouse. And then finally, the other thing that Drew and others pointed out earlier is it is primarily European adults. I'm optimistic and hopeful that the Ancestry networks are going to take a big bite out of this. And also something that someone brought up earlier, but it's largely observational. So in terms of well-characterized diseases or perturbations and other things that we would want to use for modeling or exploring, this is an area we're really focused on trying to fill these gaps and focus on how we can round out this data corpus. So one of the ways that we've envisioned doing this is really trying to work with you all, work with providers to drive costs down. And this is an example of a collaboration and a partnership with Scale Biosciences and Ultimate Genomics, in which we help support the generation of 100 million cells worth of new data. And this data is going to be generated on the order of pennies per cell, whereas a lot of those other data sets were in the kind of dollar range. And it's going to be done in about 6 months. Data will be made open. Many of the groups and folks that are involved in this, I think, are also involved in Ancestry networks and others. And so I'm really optimistic that a big chunk of this data is going to help advance the work in a couple of those deficits that we just highlighted. I'm also optimistic that there's a lot of other opportunities here. And I think we are committed and invested to drive those costs down and make these technologies more and more accessible to enable those disease applications, to enable working across cohorts and time and all the answers, so that money is not the primary limitation. And so I think working with some other reagent providers and others, there's a lot of opportunities in the future to kind of further expand on this work. Okay, and then let me finish very quickly here with coming back to disease. And this was kind of a late-breaking thing that I wanted to add in, because I know it's been on many of your minds. I was just in a breakout session, and Priscilla Chan, our co-founder, some may know, is a pediatrician by training, also an educator. And she has this wonderful quote that I think resonates and is certainly something to really think about when we start to think about the power of these technologies and the atlases for disease. And the thing that touches me about this, one, is kind of imagining being in this situation, and some of us perhaps have been, but also that even with the diagnosis, so putting undiagnosed out of the way, that diagnosis may, in many cases, be a mutation in a gene that has no known cause, and you don't even know what cell type it's expressed in. And this is something that this community is prepared to take on, and something that we also have been thinking a lot about. And a couple years ago now, so about 2 years ago, funded a couple of collaborations in which we paired rare disease advocacy organizations. So these are primarily patient-focused and patient-run organizations with experts in single-cell biology to deploy methods, spatial, dissociated, all of the things that we're talking about, against these rare diseases. And the curiosity and the hypothesis was that through the patient lens, would be able to prioritize where the technologies would be most applicable and most impactful in these patients' lives. And what I don't unfortunately have time to tell you about is that across all of these focus areas, whether it be diagnostics or biomarkers or patient stratification or new therapeutic identification, the methods and a relatively small amount of data in those methods has been transformative, and in a very short period of time. In a few cases, I will tell you, it's also a great example in which a small amount of patient data was then used to quickly fine-tune one of those large language kind of transformer-based models that I mentioned earlier, so GeneFormer or others. And from that, already starting to see some kind of quick lessons learned in terms of ways to repurpose drugs that are widely used and tolerated for these patients and for these diseases in which basically no biology is known. So this would be my kind of encouragement, or maybe sharing an area where we're thinking about what disease looks like. And the other thing that I would just say is it's maybe the inverse of thinking about kind of global burden, because by definition, these are rare. But maybe with an alternative framing instead of rare, is if you think of rare, neglected, or extremely severe disease as the place to start. And the severity of the disease, I think, has two benefits. One is very strong biological signal. And the other is that by learning those key mechanisms, could of course extrapolate it into more common diseases. And I would maybe invoke an example of like PCSK9 as something that has been extraordinarily impactful and transformative, and an opportunity to deploy atlases and human cell atlas work against rare diseases. Okay. I'll kind of quickly introduce the BioHub network. I mentioned kind of cross-institutional collaboration. The key here is to share some updates, in particular, the launching of the BioHub in Chicago and New York over the last few years, but also just to underscore that there is this theme of inflammation and inflammatory diseases, which is an area of interest to us. With that, I was going to show the example of New York, and I will thank all of you. Here is a QR code to stay up to date on our funding opportunities. I see lots of cameras coming out. That's great. We welcome those. Again, the creativity and the collaboration from this community has been really special and something that we're looking to continue with. And then also the New York BioHub, if you're interested in immune cells and immune cell engineering, is there as well. It's very new, but worth staying in touch with, and I think a lot of exciting work to come. Okay, with that, happy to take some questions, and I will also just put up my contact information as well as various CZI and BioHub networks if you want to get in touch or follow us. Thank you.
Jonah, it's really exciting to see the rare disease initiative. Would you be able to describe for the community a little bit of what are the steps on the journey from patient comes in to single cell omics suggests a diagnosis and a treatment, and how has that worked out?
Yeah, so it depends a little bit on the diseases. And so what I didn't, maybe I'll just go back. You're giving me an opportunity to explain a little bit more of this. So the selection of these groups was not entirely coincidental. So in a number of cases here, the diagnosis is made based on a mutation in a gene. So a great example of this is RUNX1, name of the gene. The mechanisms, and by and large, it's thought to be a disease that is rooted in hematopoiesis, and so hematopoietic stem cells. That is very different than some work in necrotizing enterocolitis, so a very aggressive, very early form of hypergut inflammation. And I hope that Sonia and Gary, don't chide me for getting this wrong, I think PSC Partners and primary sclerosing cholangitis is another example of a disease in which the diagnosis is one that's based more on pathology as opposed to a genetic lesion. The thing that I think is exciting is that in all of these cases, the single cell data, and after that diagnosis and the recruitment of the patients, is one in which either primary tissue biopsies can be made, but also to a question that we were discussing, circulating immune cells and others for thinking about other blood-based markers that might be useful either for disease diagnosis or for stratification is being used. And so that is the point. So downstream of diagnosis in which a lot of those patients and the samples are starting to be collected, but you could start to think about how it could inform the diagnostic odyssey for especially some of these non-genetic disorders.
Right.
Yep.
Thank you.
Great. Thank you again. Yep.
Good afternoon, everybody. I see some empty seats in the front, but I am sure you'll realize because I'm moderating this session, it's going to be one of the best sessions ever. And you should, if you're outside, come and join this session. I am Shejyuti Shaha. I'm the Deputy Executive Director of the Child Health Research Foundation in Bangladesh, and I'll be moderating this session. It's actually not the best session. It's going to be the second best session of today and tomorrow, because the best session was the breakout session that Huang and I moderated on diversity. If you missed that, you're lost, but you have this opportunity. So this is an exciting session also because we will have lightning talks, and they're going to be 3 minutes each. About 80 abstracts were submitted to be considered for talks. 12 of them were selected. 6 of them will be presented now over the next half hour, and then the remaining 6 will be presented tomorrow. I will be with you today and tomorrow. Yay! May I ask all the speakers to come up front and be ready, because we only have 3 minutes. And we will get started right now. So we will start our session with Dr. Kaile Wang. And Kaile Wang is a principal investigator at the Center for Excellence in Molecular Cell Science at the Chinese Academy of Sciences. So the floor is all yours. And there's a clock there.
Thanks. Good afternoon, everyone. It's a great honor, and thanks all the organizer to have the chance. So today I'm going to introduce two high-throughput single-cell sequencing methods we developed to detect the rare tumor subclones. So rare tumor subclones, they are important and critical, like for the tumor initiation, progression, and also the drug resistance. Like the tumor process subclones that lead to the recurrence, they are like most of the time they are low frequency, but so how to identify them is very important. So to answer this, we developed a method we call the archival nanowire sequencing, which is a high-throughput single-cell DNA sequencing method, can tolerate the FIP tissue. So use this method, you can provide around 2,000 single cells in the FIP sample. And we use this method to study the DCS to the recurrence progression, and we studied 10 pairs of primary DCS and the recurrence sample. And we did identify those persistent subclones in the recurrence. And we also found that, like in most of the cases, the primary DCS went to an evolution bottleneck model, and some of the cases went to the multi-clonal evolution. In the breast cancer field, there are another important question is what's the epithelial cell of origin of different breast cancer subtypes. So to answer this, the best way is you can identify the tumor progenitor cells and also get their genotype and phenotype information. So to do this, we develop a high-throughput single-cell DNA and attack co-profiling method we call the UDA-seq. So the DNA information can tell you the lineage of those cells, and the attack information can tell you the phenotype of those cells. So use this method, we profile a sample, have over 1,000 single cells. You can see we identify a subclone C6, which is the most early common ancestor cells. And further, we use attack-seq data to identify this C6 have the highest luminal HR epithelial model score, which suggests this ER-positive breast cancer originated from this luminal hormone response epithelial cells. And further, we found with the tumor progression, the luminal hormone response identity was kept loosened. And the other thing this method can do is we use this method to identify a lot of aneuploidy epithelial cells in the normal breast tissues. Like in this sample, we sequenced around over 1,000 single cells, and we identified 10 cells. They have a common number event. And further, we use attack information. We found those cells are from luminal circulatory epithelial cells. And another big study, which is published this month, use our archival method and UDA method. They sequenced around like 80,000 single cells, and from around 50 normal breast tissue, and they identify like 3,000 of single cells have the they identify 3,000 of aneuploidy cells, and use the co-assay information. They found those aneuploidy cells they are from luminal epithelial cells rather than basal cells. So if you have interest to our method, come to the poster 55. That's my talk. Thanks.
You were super excited. May I introduce you? So Professor Lan Jiang is going to now present. You have 3 minutes to present, and you are also from the Chinese Academy of Sciences, so thank you very much.
Thank you. Good afternoon, everyone. It's my honor to introduce my work here on behalf of Professor Zhang. Okay. Oh, sorry. Eh? Sorry. It's okay, we're not single-cell sequencing faces great challenges in large-scale clinical amplification in terms of price, sample processing approach, and multi-modality capture of the same cell. Here we developed a straightforward two-round combinatorial indexing experimental workflow named UDA-seq. It uses droplet microfluidics-based techniques for the first round of cell barcoding, followed by a second round of cell barcoding by index PCR after they are released from the droplet. Input cells or nuclei were fixed. With the post-indexing strategy, UDA-seq resulting in a 10 to 20-fold increase in the throughput compared to the standard microfluidic methods. In addition, we can smoothly incorporate this powerful strategy in the well-established droplet-based single-cell method, including single-cell core assay of RNA and commenting RNA and VDG and RNA and CRISPR perturbation. At the same sequencing depth, the data quality of UDA-seq is compared to or better than 10x genomics. The duplicate ratio is also much lower than 10x. We applied single-cell RNA and VDG UDA-seq to a frozen PBMC from human natural aging cohort and RNA-attack multi-omic UDA-seq to frozen kidney biopsy from kidney disease cohort. The increased sample size and the deeper cellular coverage enables the identification of real cell types associated with specific phenotype. Today, we have generated single-cell data from over 600 donors across 9 tissue types using UDA-seq, with over 3 million cells captured. Besides single-cell core assay of RNA and open commenting CRISPR perturbation and VDG, we also have extended the UDA-seq strategy to other multimodalities detections such as RNA and CAR-Ts, RNA and DNA methylation, RNA and RNA modification. We hope that the technical innovations will accelerate clinical translational research in single-cell technologies. Finally, sincere thanks to my PhD advisor, Professor Jiang, and my great collaborator, Huang, and other cooperators. Thanks.
Wonderful. We are sticking to time so far. So we're going to move on to our third speaker. Our third speaker is Professor Wang, and he is from the Academy of Mathematics and System Sciences, also of the Chinese Academy of Sciences.
Okay. Thank you very much for the warm introduction. Yeah, I'm delighted to share you a new method called ClusterMatch, which allows single-cell RNA-seq data in multi-scale clustering and stable matching. And we know alignment is fundamental in biology, and it aims to identify the similarity amidst the variation. And this similarity often indicated shared evolutionary history and functional relationship and the structural motif. For example, sequence alignment help us to identify the homolog sequences. And previously, we developed protein structure alignment try to optimally rotate the protein structure as a rigid body to find the similar match by a mixture of integer programming. And also, we developed some method to biological network compare alignment by integer programming, which is a much harder problem. And we are now deep in the single-cell era. So the question is how to align two single-cell RNA-seq data. And there are some challenges, specifically from the computational side. So we will need to deal with the high-dimensional noise in data. And for the single-cell clustering, it's intrinsic multi-scale, and we need to determine the optimal clustering resolution. And also, it's very difficult to interpret the gene program. We propose some ideas to overcome those challenges, and mainly regarding to the multi-clustering and also the stable match. By putting these ideas together, we propose this new method called the ClusterMatch. It takes two single-cell RNA-seq data as the input, and then we output the cluster alignment graph. This graph can align two data sets at the cell type, cell cluster level, and it can borrow information from each other and to determine the optimal resolution for the clustering. And it also can align the cell, giving the marker gene and enhance the interpretation. And this leads to a nonlinear, non-convex optimization problem. We solve this one by a heuristic algorithm. And we validate this one in a live human and mouse pancreas single-cell set. It can reveal the cell type both at cell type level and the subtype level. And those clusters are validated by the manual annotation. And our method can improve the clustering resolution compared to the state-of-the-art cluster tree method. And it can better remove the batch effect than several SOTA method. And it can improve the cell type annotation accuracy better than the CRAT. Also, we can compare the sheep and cattle single-cell RNA-seq data and to find a conserved cell type during the rumen development. And the method is published in Bioinformatics, and also the software is available in GitHub. And we prepare a very small poster, and please visit. Thank you.
Thank you. We'll move on to our fourth speaker, Ms. Rongting Huang from University of Hong Kong.
Good afternoon, everyone. I'm delighted to share the research work about the comprehensive benchmarking of the copy number alteration analysis methods from the single-cell and the spatial transcriptomics data. As we all know that the copy number alteration have been widely shown to have associations with the various cancers. And the high-throughput transcriptomics data provides a way to detect the copy number alteration at the single-cell resolution. However, it is very challenging to detect it accurately, so it is crucial to systematically evaluate the performance of the leading single-cell RNA sequencing copy number alteration detection methods. Here we select the 5 leading methods: InfraCNV, copycat, Casper, numbite, and scClone to benchmark on a glioma sample with the CNA profile characterized by the WGS as a very good ground truth. And here in this glioma sample, there is a chromosome 14 allele-specific copy loss in different subclones. And the NumBite and the Casper failed to detect this kind of event. However, the X-clone can detect copy gain, copy loss, and loss of heterozygosity quite well in all CNA states. And I extend the 5 single-cell RNA sequencing detection methods to the spatial transcriptomics data within liver sample with 3 sections in normal, transition, and the tumor state. Here, X-clone can also detect a very unique signal in the transition section in the chromosome 1q, with, as shown in the WES ground truth. However, other methods cannot detect this kind of information, which also provide information that the ST data can be used for the allelic copy number alteration analysis. Here we simulated more CNA states in the liver spatial transcriptomics data, and the copy number, the CNA ground truth of this simulated data set shown in the panel A here. And we can see that after apply all of the methods to this simulated data set, the CNA pattern applied to the spatial context can have a very clear pattern of the subclonal information. And it is also clear that the chromosome-specific CNA state can be obviously visualized in the spatial context. We know that the spatial transcriptomics, especially the Visum version 1, is similar with the single-cell RNA sequencing data. However, it still has a more sparse. It is more sparse and also has the mixed cells at the spore level. So it needs more efforts to extend from the single-cell RNA sequencing to the spatial transcriptomics data. And based on the trials of the application of these methods, I summarized the characteristics of the methods. Hope it can be a good guidance for you to select a proper method for your specific data set. Thank you, and looking forward to more discussion during the poster section. Thank you.
Thanks, Rongting. Next, we have Ms. Jean Lee, and she is a graduate student at the Seoul University, National University College of Medicine.
Thank you for the introduction. I'm Jean Lee from Seoul National University, and I'm truly honored to present our findings today on generic dissection of Hershey-Sprung disease pathophysiology. Hershey-Sprung disease is a devastating condition characterized by the absence of anterior ganglia in the distal ball, leading to severe digestive issues. While LET is a major gene identified to us, and mutations in LET are linked to Hershey-Sprung, but not all patients carry these mutations. We've categorized Hershey-Sprung into 3 subtypes for our analysis. First, typical HD, the most prevalent case in Hershey-Sprung. Second, the hypoganglionosis are characterized by significantly reduced number of glial cells throughout the colon. Third, total colonic ganglionosis identified an absence of glial cell in total colon. To investigate this variation, we conducted single nuclear RNA sequencing on 13 Hershey-Sprung and 3 control tissues. This slide displays UMAP plots segmented by Hershey-Sprung subtype, and our analysis indicates altered cell compositions in Hershey-Sprung, specifically reductions in muscles, smooth muscles, and interstitial and neural cells with increases in B and T cells observed in the TCA. Our gene expression studies of glial and smooth muscle cells revealed the differential expressed gene linked to muscle contraction with notable downregulation in the TCA. Furthermore, we found significant downregulation in neuronal cells of hypoganglionosis. PSI analysis revealed collagen-related changes across Hershey-Sprung subtypes. Additionally, gene ontology analysis of germinal center of B cells highlighted a rise in cell cycle-related terms, particularly in the TCA and typical ITD, a pattern also supported by cell cycle scores. We identified differential regulon activity across the Hershey-Sprung subtype, and in our study, approximately 40% of samples harbored a lead mutation. We are continuing our analysis and include more data to further deepen our findings. In summary, our single RNA-seq has revealed significant insight into the genetic landscape of Hershey-Sprung influenced by subtype. I would like to express my gratitude to my advisor, Professor Choi, and at SNUH, Professor Kim and Go, and my lab colleagues. Thank you for your attention.
Thank you, Jean Lee. And Jean Lee actually gave us 42 seconds back. Last but not the least, we have Dr. Yip-Chi Wai, and he is a research scientist at RIKEN.
Hi, everyone. I'm Morris from RIKEN, and today I will share with you my recent work on transcription factors and their distinct modes on enhancer and promoters. So in a differentiating time course, we have IPS, NSC, and quantico neuron. These cells are subjected to single-cell 5'N RNA-seq and also single-cell ATAC-seq. So after co-embedding, the single cell will group into metacells, and each metacell will contain both ATAC and RNA signal. The cis-regulatory element, CRE, can actually quantify by both RNA signal and ATAC signal. So it is what we call bimodal CRE. And we also use these two signals to get the TF motif activity. So finally, we have 4 different data sets on all the metacells, focusing on the CRE and the TF motif. And of course, we also have the expression level from the single-cell RNA-seq. So to annotate the accommodating state of each of the CRE, we also perform cut and tag for a couple of histoma. So now we also know that this CRE are promoter or enhancer, active or repressed. So after we load the enhancer or promoter of each CRE, we look into the correlation of the TF expression and motif activity. So the first thing we find that is the chromatin of enhancer is actually more commonly regulated by TF compared to promoter, according to ATAC signal. Versus we look at the TF that regulate the transcription of promoter, they are actually very unique from all the other TF. So if you look into the breakdown of each individual TF, the first one is actually OFF4. You can see the OFF4 is regulating the chromatin signal of the enhancer only, but not the promoter. And at the same time, in the lower panel, you can see it also doesn't change the RNA signal of any promoter or enhancers. Versus at the very far end, this SOS3, it is a repressor, so you can see that it doesn't change the ATAC signal, but it represses the RNA transcription from the promoter. So to validate this observation, we use ChIP-seq and knockdown followed by ATAC-seq on TAD and also one cut. So you can see all these affected CRE with chromatin remodeling. We can see most of them, or majority of them, are enhancers. So finally, we also use this bimodal CRE to build a GRN, which is much more comprehensive than before. So in the example, you can see that besides, we can identify the relationship between TF enhancer and promoter, we can also see which kind of signal transcription or commutative ability drive the interaction. Finally, I want to thank all my coworkers and collaborators from Phantom for their input in this project, and also thank you for your attention.
Thank you, Dr. Ip. Thank you to all our 6 speakers for being on time. Another round of applause for all the 6 speakers, please. This session is done. I'm not leaving yet. So we're going to move on directly to bring back our discussion points, what we learned in our breakout sessions. And we're going to obviously start with the best breakout session, right? The one on diversity. I was the comedian there, and Kian Hong was more the moderator. So Kian Hong is going to summarize all the things that we discussed and learned during our breakout session.
Great. Thank you, Sanjuti. As Sanjuti mentioned, we had the privilege and pleasure of organizing or co-chairing one of the best breakout session. I think it was the first time we really had every single attendee contribute and speak up. So we wanted to start first by thanking the attendees for their enthusiasm and their engagement. So we focused on 3 questions in this session, and we've grouped the first 2 together. So one is, what are resources that would be helpful for the diversification of cohorts across Asia? And the second is, what capabilities would we need to build to advance single-cell genomics, especially for communities that have been traditionally underrepresented in research? So one of the first points that was brought up was really the importance of community engagement and also meeting local needs. So it's not, I think we have to avoid having this colonial mindset, especially when it comes to underrepresented communities. So it really needs to start community engagement right from project design and throughout project implementation, and to understand what the community needs. So for example, we heard from attendees about, say, when you speak hunter-gatherers, maybe they don't need fancy healthcare resources, but they really need things that are targeted to their environments, like forest management, protection of environments. And also one of the things that was brought up was that when we're conducting single-cell studies of these human cohorts, can we bring about clinical benefits to diagnostics and healthcare together with the metadata that we're collecting? So a lot of our projects collect PAD, clinical metadata, blood biochemistry, for example, and can we return some of these findings to the community and also pair this with the research that we're doing in addition to the single-cell profiling? And one additional example I wanted to bring up over here is that, for example, when to tie this in with some of the goals for the HCA in terms of the diversity task force recommendations. So we spoke about blood being one of the most accessible tissues, and then nasal swabs and perhaps skin being the next most accessible. So being able to say pan-nasal swabs with rapid diagnostics for pandemic-related diseases or for pandemic surveillance. So that's a really direct means of being able to bring about benefit to local communities while pairing this with research. So I think community engagement is key. We spoke about capacity building and training, so about workshops, bi-directional travel, being able to bring experts to the community and to bring researchers to labs that are well-established in these single-cell experimental and analytical methods. And also being able to have one-to-one training and to develop trainees, train PhDs through workshops, as well as more in-depth training embedded in the home labs or bringing experts to the communities. So we also spoke about being able to share our knowledge bank of the community. So, Sanjuti has had a cohort that's been built for 20 years in Bangladesh, and there have been many examples of HCA cohorts, including IDA. So we were saying that, can we share insights? So what are some of our donor inclusion-exclusion criteria that we can benefit, that can help to educate and bring about insights to other cohorts? So in thinking about stuff like healthy donors versus population baselines, I think very fundamental project design parameters. It's also important to think about ethical guidelines in the context of really local sensitivities and local legislation. So there are some communities where some metadata fields are sensitive, so reporting self-reported ethnicity metadata for refugees or for immigrants, for example, can expose them to potential of stigmatization or discrimination. So I think there needs to be local sensitivity when you're reporting or collecting such metadata. And also thinking about local legislation. So I think, in mainland China, there are challenges to data sharing, I think because of legal complications. And, we heard, we would love for further engagement, I think, with Chinese researchers in the human cell atlas community, but there are roadblocks to data sharing. So thinking about how we can better communicate these to local authorities and to explain some of the benefits of research and to place, I think, what we are studying into context and to be careful about data sharing, respecting local legislation while being able to maximize benefit. And finally, I think there's a HCA section on protocols.io, for example. So there's been efforts to do protocol development, but many of these occur in well-resourced settings. So it's almost like the perfect environment for developing protocols. But it's very hard to do this in the field, and can we meet people halfway? So what are people actually doing in practice in under-resourced settings, and can we meet them with single-cell profiling methods? So that's really the first set of discussion points. And then we touched upon disease, and I know there's a disease breakout session, and Jay and Piero will be talking a bit more. But I think this is where we got all of the attendees to be able to talk about diseases of interest, and we heard about personal stories, how some of these diseases actually impacted their parents or their grandparents or one side of their family. So I think a lot of importance, personal importance, and also societal importance to them. We heard about infectious diseases and associated complications, lifestyle-related diseases, neuropsychiatric cancer, etc. There were some of the possible priorities, but we wanted to juxtapose this against the HCA short-term and long-term diversity task force goals. So in the short term, we wanted to bring, say, blood to 50 countries because it's really one of the most accessible tissues, and it's almost like a bridge to be able to bring about additional tissues and single-cell research in more challenging sampling sites across global communities. So, for example, perhaps you were able to think about infectious diseases first, but also in parallel launch capacity building and community engagement efforts in order to be able to study cardiovascular disease, for example, in harder sampling sites. And last but not least, I think there was a strong interest in matchmaking people with similar interests. So it's great we have got opportunities like this to talk to people, but this needs to happen more often. So how can we best bring together collaborations, foster collaborations and communications, and matchmake experimentalists with clinicians, with analysts and bioinformatics. So I think these, and we're interested in continuing this conversation. So we've got a poster at D134. We've got a QR code that you can scan there to give your feedback, and we're here till tomorrow. So please come and chat to Sanjuti and us and Jiang, and we're happy to keep the conversation going. So thank you.
Okay, we had a great spatial session as well, where Al Forrest and myself facilitating. Right. So part of this slide is stolen from Holger Hein's summary presentation at the HCA general meeting in Milan. So if his slide is bad, it's all his fault. Well, not much to say. The key thing to say here is that we're ready for prime time. Spatial technology is ready for prime time. There's many technical challenges. We discussed almost none of them. We discussed what I'll show in the next slide. But we can come back to this later. There's only two more slides here. One slide this and one Al is going to show. If anyone has any questions or suggestions or ideas, constructive ideas on this topic, please raise your hand while we're talking, and then we'll talk fast and leave time for Q&A. If no one raises their hand, we'll talk slowly and use up the full 6 minutes. So what we focused on was building a global consortium. The timer wasn't turned on, so I have lots of time. So, how do we build a global consortium, and how does HCA contribute to this global consortium? And so if we build a new spatial project within HCA, HCA Asia, what boxes does it have to be check? Or what boxes does it have to check? It should be diverse. It should be not just Asia. That's one. Then it should have a disease focus, either high disease burden or, as Jonah was talking about, an underserved rare disease where there's a clear benefit you can show, and that's the whole point. We want to be able to show benefit from this in terms of diagnostic drug targets or repurposing existing drugs for rare disease, for example. Then it has to be complementary, of course, with existing initiatives, because there's so much to do. Why do something somebody else is already doing in this field? And then spatial molecular profiling of some accessible tissue has to be relevant for that particular disease, biologically relevant, and it has to build on existing capabilities in the HCA, our technical capabilities, our data analytical capabilities, and perhaps above all, tissue availability within our network, which Al will come to next. And so based on all of these criteria, if we want to check all of these boxes, which organs and diseases do we prioritize for spatial omics was the question. Over to you, Al.
Okay, so this is a rough description of what we discussed. So we were talking about project probably on FFPE archival tissue, because that would enable broad access to disease samples. We had a show of hands, so if we were going to do something, what disease should it be? There were about 10 people put their hands up for cancer, 2 for genetic disease, 0 for infection in this particular audience for this meeting, that little breakout session, and 1 on cardiovascular. We talked about FFPE on cancer and what sort of cancers would you focus on? Would you choose one or would you choose multiple? And it was suggested that we should focus on cancers with different prevalences, outcomes, genetics or etiologies in Asia. For example, colorectal, oral, ovarian, breast and liver all have some sort of different properties in Asian populations. Microbiome was also mentioned. We discussed about how to access match normal. So what sort of normal is sufficient for these studies? Can it be from a different donor? Can it be transplant? Or is normal adjacent to tumour? Normal adjacent to tumour has the advantage that it's got the same genetics, but it's not really true normal. There was also, as always, funding concerns. So how do you fund these sort of initiatives? So normal is really hard to get funded nationally in many countries. Inclusion of disease makes it a lot easier to get funded in countries. Multinational consortium could help us attract international funding, including philanthropy, and that would help us with the normal. And if we're going to focus on, say, 5 cancers, not 1 cancer, how many samples would you do? How many countries would you, how big does it need to be to show that you've got good coverage? And then I sort of tongue-in-cheek added, is spatial really ready for cohort study? So how many patients are you going to have to do with spatial to have the power to actually say something meaningful? And then lastly, we talked about how to deposit and share spatial data. So if anyone's got any questions, please follow it up via email to 2 of us, or if you've got any comments now, go for it. We've got like 0 seconds left, apparently.
All right, I should introduce the next 2 speakers then, or the next one speaker, John Randall, Chief Alliance Officer for the Human Cell Atlas.
Thank you, there's a comment.
Thank you very much, Jiang. So in this breakout session, Chloe Villani and I led a discussion about challenges and barriers to data sharing. We were interested, of course, being an HCA meeting, specifically in challenges for sharing data with and submitting data to HCA, as well as downloading HCA data from the HCA data repository, and also from Cell by Gene. I will say that, yes, sorry, I need to advance the slides. So I will say on point 2, actually, we really didn't hear too many challenges, and that was really encouraging. It also really comports with the data that we have on the data access side, because we, for example, just from the APAC region, the Asia Pacific region, we see typically about 20 to 40% of our download requests in any given month are coming from this region. And so that's really encouraging. That being said, if you have encountered challenges in downloading HCA data, please let me know or anybody in the executive office and we'll get it straightened out. So we really focused on the challenges for sending your data to HCA. And for this, I just want to remind you of what you heard this morning, that we really use 3 different resources for sharing different categories of data and metadata. You heard about in the center there that cell by gene is really what we think of as our matrix store, where expression matrices and that tier 1 metadata, largely technical and project metadata, are stored. The HCA data repository at the bottom can store FASTQ files and also tier 2 or biological or diversity or medical metadata. And that can be under managed access or open access. So that's really for any sensitive data, it's really the HCA data repository that we need to use to store that. And then at the top, you heard about the cell annotation platform as well that has the cell annotation metadata. I just want to remind you that despite the fact that this is this tripart structure, this is all coordinated very well behind the scenes and hopefully is very seamless for you, the data contributor. And then the Atlas assembly team can then use data from any of those 3 sources. And as well, that can be made available through the HCA data portal. So I already mentioned that managed access really is that bottom data repository. What we have not talked about in this meeting, but I want you to be aware of in brief, is that in parallel with that sort of technical data flow, HCA has also put in place over the last few years a number of legal contracts that enable us to handle this data responsibly and in accordance with your expectations as a data contributor. I'm not really going to go through this except to say that in purple is the data flow. So there are 3 institutions, EBI Broad, the Broad Institute, and UCSC that sort of operate the HCA data repository. If you're a data contributor, you and your institution will sign a contract with HCA, and that contract both sort of specifies that you have the proper consents and the permission to share the data with HCA. It also sort of enables us to have a mutual understanding about how your data will be stored and accessed, and that would include open access versus managed access. And the key for this is that all these arrows are legal agreements that ensure that your expectations as a data contributor and the terms of the consents that you have are passed throughout the system to the people who operate the data repository and ultimately on the right to the data user. So that data user also has to sign a contract with HCA that binds them to these terms as well. So this is very complicated, but it basically says that this is the system by which we handle your data responsibly. So getting back to the breakout groups, we examined a number of issues. The first issue that came up was, well, what happens if I've already submitted my data to a different repository, or if I'm required by my funder or a national government to submit data to a particular other repository outside of HCA? And I want to assure everybody, not only is that permissible, but that's actually quite common. People will submit their data to like EGA, ENA or something, but it's also in the HCA data repository. And as you heard, the reason that's important is that in the HCA data repository, we can ensure that the metadata and the file formats are consistent with HCA standards so that we can actually integrate your data sets into atlases. So secondly, we were asked about other data modalities and specifically things like attack-seq. This is where the HCA data portal is designed with a lot of flexibility. So we can link out to other repositories where you may end up sending those other modalities of data. So attack-seq is actually a very good example because in one of our published atlases, which is the Retina Atlas from the iBio network, they actually use the UCSC cell browser for their attack-seq data, and then we actually have a link out to that to those files on the HCA data portal. So the HCA data portal is very flexible. It allows us to link not just to cell by gene, but also to other repositories as needed. As I already said, we didn't hear about data barriers to accessing data, but let me know if there are any. And finally, a point that Huang and that first breakout session already brought up is there are cases where we understand that there are barriers to sharing FASTQs outside of borders, particularly FASTQs. I think the key point here is that even if that's the case, we still want to have a conversation with you about sharing your expression data because that may be possible. And furthermore, we can actually share the code that we use to generate matrices from FASTQs. And so you can actually keep your FASTQs locally, but generate matrices that can be shared and then used in the Atlas assembly process. So we have done a lot of work over the years, and I do want to say that a lot of the work, both the data ecosystem itself, particularly the HCA data repository, has been supported by CZI, but CZI also has funded all of this work that has led to the legal agreements and the managed access system that enable us to responsibly hold sensitive data within HCA. But even if you can't, even with all of that in place, if you still can't share your FASTs with us, we want to have a conversation about expression data as well. So I'll end there, and I'm very pleased to introduce the leaders of the next discussion, which is Piero and Jay. We have heard a lot already about new potential disease directions within HCA, and they will explain to you what the breakout group had to say about that.
Thank you, John. Actually, I think we had the best breakout session. So. Yes, save the best for last.
We are very competitive, okay?
Yes, and actually you guys said everything we talked about, so I think we're done. Because you guys talked about disease, and Spatial talked about disease, and we actually talked about disease. So to simplify, we just made a picture. So picture speaks 1,000 words. To summarize what we discussed, of course there's a lot more, but just to summarize the fact that, of course, disease is not categorical, and it's one big understanding. We have to understand that it's continuum, and with that notion, it comes with complexity and the size and the metadata. All of that needs to be extremely well recorded, and therefore, how to best stratify? Should we even stratify these data across the spectrum of disease? And so there's something that we have to be aware of. Of course, normal sample, healthy sample is also not so clear-cut. It's not necessarily a categorical either. So these are something that we should always be mindful of. But of course, there are broader categories. It could be infectious disease, inflammation, chronic inflammation, cancer, metabolism, aging has been mentioned. But of course, coming down to practicality, which of these samples are most accessible and most relevant in Asia and most impactful for therapeutics or diagnostics, and also ease of sample collection. So here is just a very small snapshot. PBMCs are, I think, still very much relevant in context of inflammation and infectious disease. And I think there should be a continual support for this, and also nasal cavity samples are relatively easy to collect. And there are now well-established protocols to do this. And so in a way, we can achieve scale, diversity, and we already have know-how to do this. So these are kind of low-hanging fruit for us to do. But on the bottom left, you have clusters of cancer types, largely in FFPE collections. Probably the scale will be much smaller, somewhat accessible, and whether they're shareable across different countries is another issue, maybe not so straightforward. But what we talked about is there are also other consortiums out there, like KPMP or Tumor Network, Human Tumor Atlas. And so what are some of the things that we can learn from them, and what are some of the things that we can be differentiating or also be synergistic? And so I think we should be more proactive in reaching out to other consortiums and other foundations, like Hemsley has been doing IBD of the colon. So what are the things that they've learned, and how we can co-develop with them in some ways. I think, for example, like metadata collection and what are the key important clinical information that need, things that would be extremely helpful. And of course, put skin there, it's kind of a bit biased. There's a skin breakout session tomorrow. It's kind of in the middle ground. I think it's kind of relatively easy to access, yet there are extremely wide range of disease, including atopic dermatitis or psoriasis, and a great sample to study in spatial context. And we'd love to hear some of you what your thoughts are, because I've already discussed with a few friends and colleagues in Japan and in Korea that they already have banks of skin samples in FFPE, so it's ready to go. But also there will be a breakout session tomorrow specifically on infectious disease. So if you're interested in that, please join. And there will be a breakout session on cancer as well. So if you're interested in cancer, please go and join those breakout sessions. And on the right-hand side, we talked about, should we have large number of samples for one disease type, or should we have many different disease types for one sample size? I think at an initial stage, we thought it would be good to just have focus on 1 or 2 disease types and go as wide as possible as a pilot. But again, we would love to hear what you think.
Jay, you summarized perfectly, and I don't have anything. It's the perfect. It's the perfect summary of the perfect presentation. Questions?
Yeah. So we have 2 minutes. I intentionally made it short, so I want to hear from you. I have to say, we were also quite active. A lot of people spoke out, and they shared these information with us, so thank you for your participation. And anyone else? Okay. Yes, Sham, and Chloe, and yeah.
Okay, quickly, did any disease stand out as, this is the winner in terms of broad interest?
No.
All right. We'll keep working on that.
Yes. And thus, I may have to make this chart. Yes, I think there was a wide range of topics that were raised.
Jay, I was hoping you would show the QR code for the questionnaire. So perhaps you can let people know that we're circulating a questionnaire to gather the information about the diseases of interest and samples that we have. You want to expand on this?
Yes. Has that been sent out? Yeah,
it was there this morning.
Correct.
But we're gonna maybe we bring it up again tomorrow.
There you go.
There you go.
Thank you. So there is now a survey that will ask you what kind of disease samples you're working on, and what's the sample size, and what are some of the possibilities of sharing. And this survey can also help for us to connect you to the right group of people so that we can co-develop this. And I think this will be sending it out to broader HCA community, of course. But you guys, HCA Asia has the first dip, I think, right? Like this survey was just made last night. So please participate, and we'd love to hear from you. Correct. And Chloe says that I should say that it will also help you to prioritize, to define, and it will also help us to prioritize which disease to go for the global HCA effort. Okay. All right.
Thank you.
Thank you. Angela.
Cool. All right. I'm back to wrap up the talks, at least. Can we get the slides up? Okay, so that was what happened today. Actually, after this, so all the talks are done, but we do have the poster sessions. A lot of attendees have prepared really nice posters. I haven't had a chance to look at them, so this is a great opportunity to interact with them. Some of the short talks you heard, they have posters. Some of the people who didn't get a chance to talk, they also have wonderful posters. So please do stay for the poster session. And then after that, there is a change in the timing of the shuttle. So in the morning, we said that the shuttle was leaving at 7:10. It is now leaving at 6:30 from here. Okay, 6:30. So, Sorry. In the morning, the slide showed that the shuttle would leave at 7:10. The time has been adjusted. So the shuttle will leave at 6:30, which is an hour and a half from now, roughly. So make sure you catch the shuttle if you want to go back. And yeah, I just wanted to say thank you to all the speakers. Really wonderful talks. I really enjoyed it. I was part of the diversity breakout session. I declare that that was the best session. And yeah, I hope you guys enjoy the poster session, and we'll see everyone back here tomorrow as well. Thank you.